So Dr. Strick's research focuses on 4 major areas: the generation and control of voluntary movement by the motor areas of the cerebral cortex, the motor and cognitive functions of the basal ganglia and cerebellum, the neural basis for the mind-body connection, and unraveling the complex neural networks that comprise the central nervous system. If you're anything like me, that means you're going to have a lot of questions because I don't know what some of those words even mean. Dr. Strick received his BA in biology and PhD in neuroanatomy and neuroscience from the University of Pennsylvania. Before moving to the University of Pittsburgh, he served for 24 years at the VA Medical Center at Syracuse, New York, and SUNY Upstate Medical Center, and he's earned many honors. Serves on numerous prestigious associations. He served on many national and international committees. But rather than forcing you to listen to me list these honors, I suggest you read the article about him in today's Daily and that we let him address the topic of today, your brain in action. Please welcome Dr. Peter Strick.
Well, I've often wanted my sons to hear those introduction remarks. They see me as a tired old basketball player. It's a real treat to be here. The notion that I get to be at this institution, to see the environment, free food and free lodging, and all I have to do is give a 40-minute talk, it's just amazing. So I want to give you a perspective about the brain. And it may be a little different than what you normally hear when people talk about the brain. The first thing I want to do is give you a demonstration of the brain in action. Now, some of you have heard that an old dog can't learn new tricks. And what I want to do is demonstrate that you can learn at any age. And so I've identified a volunteer, if you'll come up. So the first thing I want you to do is just stand right here and close your eyes and watch what I'm going to do. I'm going to show people my sense of straight ahead. Okay? So hold up your arm, close your eyes, and straighten your arm and show them your sense of straight ahead. Okay? Everybody can see that, right? Pretty good. Now take a step back. Okay. I'm going to hold up my finger. You can open your eyes, drop your arm, and touch my finger as fast as possible. Faster. Faster still. Okay, just go straight at it. Way to go. Okay, now we're going to put on some prism glasses. And they shift what he sees from what he feels. Okay? So just once, touch my finger. Fast as possible. Okay, everybody saw, he missed. All right, we're going to do it one more time. All right, now, your brain is going to take care of this. I don't want you to correct. I want you to point to my finger where you see it as fast as possible. Okay, so he missed by about that much. All right, so I'm going to let him practice. And his cerebellum, it's a repair shop in the brain, is going to adapt his motor output and also adapt his sensation. So after about 20 trials, he's going to start hitting my finger. All right? So I'm going to fundamentally change his brain. And this is adaptive plasticity and motor learning. So what I want you to do is still, fast as possible, point where you see it. Keep going. Keep going. Okay. So it took about 5 trials. Keep it up. Yeah, okay. So you're trying to do a cognitive correction. That won't work, okay? No, you got to let your cerebellum do the job, all right? Trust it, okay? Keep pointing, fast as possible, fast as possible. Keep going, keep going. Okay, so still you're doing a cognitive correction. Your cerebellum will do the job, okay? Touch it fast as possible. There you go, again. Again. Keep going. Very good. Okay, so he's about on target. His cerebellum has adapted his motor output. Now, I want you to do it with your left hand once. Whoa! Everybody see that? I only screwed up half of his brain, okay? So what he did was miss by a mile, all right? So with your right hand, touch it, okay? And now with your left hand, no, see, he's off by that much, all right? So you only adapt what you use. Every time you move, your cerebellum looks at your intentions and then the actual output. And if there's an error, it corrects that error, and it does that gradually. All right. Fast as possible with your right hand.
Right hand.
Yep. Keep going. Okay, you hit it. Okay, left hand. Okay, so I'm only screwing up half of his brain. And you think, don't we generalize? Don't we learn with one arm and transfer to the other arm? And the answer is no. The only way that you learn is by using something. So I'll show you how that's true. You haven't practiced over here? Touch as fast as possible. Okay, you're off by that much.
Touched it.
No, on the second try, okay? Fast as possible. Yeah, right? Okay? So he didn't adapt over here, all right? So he practiced over here, and that's where he's adapted his motor output to meet his sensory input. But he didn't adapt over here, and he didn't adapt with his other arm. So learning in this case is learning what you practice. And practice makes perfect. Okay, so touch this as fast as possible. You haven't fully adapted. Now you're doing it. Okay, fast as possible. Okay, now this is permanent, all right? Take off the glasses. Okay. You play golf or anything?
No.
Good. Okay. Fast as possible. Whoa. Everybody see that? He was off the other way. So I'll do it one more time. Stand over here. Okay? Fast as possible. Whoa. No. This is permanent. Okay? It's absolutely permanent. This will last as long as he doesn't use his arm. Okay? So we're going to have a demonstration of that. Okay? Put the glasses back on. Okay, practice hitting my finger. Okay, now go sit down. Okay. And at the end of the, take your glasses off. Okay? So hopefully what I've convinced you of is that old dogs and also young people and you, your brain has adaptive plasticity. And practice makes perfect, but what you don't practice doesn't change. So I'll have him come up at the end of the talk, and you'll see that without the glasses, what he learned stays. And the only way that he'll adapt back to his normal input-output relationship is by practicing. So let me move to another topic, the mind-the-body connection. When I was a kid, I would get stomach aches in elementary school. And they'd occur before big exams, they'd occur before little exams. And my parents took me to a doctor, and the doctor said that he couldn't find anything wrong with me, that my stomach aches were psychological in origin. Well, what does that mean? You know, when somebody says you have something that is psychosomatic, it used to mean, and as a kid what I took it to mean was that I was a whack job, that I was a psycho. That basically this was imaginary, it was all in my head. It wasn't real. I can tell you the pain was real. As I grew older, I stopped having stomach aches, but I had migraines. And I learned, I had a migraine whenever I was in a situation where I felt trapped. And so I learned how to get out of those traps. But the migraines were real, and some of you may have migraines, some of you may have stomach aches. It turns out that family physicians will tell you that about 30% of the patients who come to them have medically unexplained symptoms. The symptoms are entirely real, but they're judged to be psychogenic, psychological, psychosomatic. There are disorders like essential hypertension. What is essential hypertension? Well, it means essentially the physician doesn't know the cause of your hypertension. So I developed a technique which allowed me to look at neural pathways. And it uses rabies virus. And we did experiments in non-human primates to look at the regions of the cortex that control internal organs. So we could look at this brain-body connection. Now, we did the experiments in monkeys because we wanted to do something that was relevant to the human condition. You may not know this, but 94 to 96% of the neuroscience experiments are performed in rodents. Now, I give a lecture, and it's why rodents can't play the violins. And it's not because we lack small violins. There's a wonderful artist in the Netherlands who makes small musical instruments for mice. But I'm sure you guys in the audience recognize that there's a difference between rats and humans. Rats don't write symphonies, don't write novels. Our brain is about 800 times larger than that of a rodent. And the difference is that there are areas of our brain that have been added that rodents don't have. And so some of what I'm going to talk about is possible only because we did these experiments in monkeys. So we injected this special tracer into the adrenal medulla because we're interested in the top-down control of stress. And the adrenal medulla is one of those organs that is really sort of the first responder, fight or flight. Somebody insults you and you turn red, get angry. It's the adrenal medulla that's the first response. And what we wanted to understand was what regions of the cortex, through multisynaptic connections, influence your adrenal medulla. And the results really amazed me. One of the regions that was very important in controlling the adrenal medulla was the primary motor cortex, the area that you use to control your arm and your legs and your proximal body muscles. And when we looked at what part of the motor cortex is a map. It has an arm area and a face area and a leg area and an axial body representation. And most of the input to the adrenal medulla came from the axial body representation. This means that your core muscles have something to do with the control of stress. Now, my sons have been telling me for years that I should do Pilates or yoga, that I should do something to control stress. And I said, this is ridiculous. There is no neural explanation why yoga could have any impact on me. This is just nuts and granola. But here I find out that the region of the cortex that has the densest, the most control over a stress organ is axial body representation. So I actually started doing Pilates. The other region is an area that is active during what's called cognitive control tasks. So when you make a decision, when you sense conflict, this area lights up. So does it surprise you that when you sense conflict, you control your adrenal medulla for stress responses? Now, what kind of conflict are we talking about? Well, I'm going to do a demonstration for the people in the audience where we demonstrate subtle conflict. It's called the Stroop task. So everyone can read this. The word is red, and for those of you who are like me, who are colorblind, it's colored blue. So the word, if I say the word, you say red. If I say name the color, you say blue. And then we have this one, okay? It says The word is blue, and it's colored red. Okay, now I'm going to flash these up and just say word, color, and I'd like everybody in the audience to yell out, red, blue, all right? Pretty simple, right? Okay, you ready? Word. 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 Color. Color. Word. Color. Aha! Okay, so we created a mild conflict, all right? Well, it turns out that for 10% of the population, that mild conflict causes a spike in blood pressure that for those 10%, it's predictive of cardiovascular disease. So I don't do this demonstration very often or for very long because I don't want to hurt anyone in the audience. But it's your regions that are involved in cognitive control. Even mild conflict is enough to set off your adrenal medulla. Now, another region that we saw that goes to the adrenal medulla is an area that lights up during mindful meditation. Now, here again, my sons have been telling me, Dad, you ought to try transcendental meditation, mindful meditation. And I've said, give me a break. I don't have enough time in the day. I walk the dog, take out the trash. I don't have time for mindful meditation. And there's no neural substrate. There's no way that mindful meditation is going to turn on circuits that have some control over my adrenal medulla. But my sons were right again. There is a region of the brain that is active during mindful meditation, that has a relatively direct connection to your adrenal medulla. I have not started mindful meditation yet, but I highly recommend it. Now, you say, look, can thinking about things, can engaging your cortex cure cancer? I'm not saying anything close to that. But I am saying that you have cortical areas that control your internal organs. And that control can influence your immune system, it can influence your cardiovascular system, it can influence your gastrointestinal system, and can influence stress responses. And we don't know very much about this. So now when somebody says you've got something that's psychosomatic, you can say, yeah, it's all in my head. And your brain is in your head. And it's real. So this top-down control, the brain control of internal organs is something that's real. There are real neural pathways that mediate this. Okay, my third topic. So I don't often get to talk to hundreds of people about science. And so this is my chance to tell you something about the process of science and give you an example that illustrates one of the problems that we have today. So I play the lottery whenever I think that the amount has gotten large enough so that if I win, it would substantially change my life. And my sons tell me, I have very bright sons, they tell me, Dad, your chances of winning the lottery are the same as your becoming Miss America. And I've said, my chances are a little bit better than that. But I don't have a chance to win if I don't buy a ticket. And that's a description of the status of research in the United States today. The funding at NIH is at all-time low levels. We're not able to buy a ticket. So if you look at what's happened in the field of cancer, it's really remarkable. My mother died at the age of 43 of breast cancer. My wife and sister are breast cancer survivors. It used to be if someone was diagnosed with a melanoma, it was a death sentence. Now, many of us will know people who are either well-maintained or in recovery. I have a brother-in-law and another sister who had lung cancer, and they're survivors. You can't say that about neurologic disease. We discovered the gene for Huntington's disease more than 30 years ago. There's no change in treatments for Huntington's disease. In the 1960s, we discovered the region of the brain that degenerates first in Parkinson's disease. We have treatments for Parkinson's disease, but they don't slow the progression of the disorder. Lou Gehrig's disease, amyotrophic lateral sclerosis, there are no better treatments today than when Lou Gehrig was first diagnosed. My nephew died of a glioblastoma more than 20 years ago. The treatments for that are no better than when he died. So the progress in neurosciences in those areas in terms of treating disease is well behind other areas. So it's really critical to buy a ticket, and we're not able to do that. Today, less than 10% of submitted grants are funded. And so what we're losing is the opportunity for discovery. And that's a really critical opportunity. The grant mechanisms today are only for those projects that are hypothesis-driven. But what does that mean? Well, that means that you have a clue in advance that you're testing that something is right or wrong, and it often leaves out the opportunity to discover something new and unexpected. So I want to give you an example of discovery. So a colleague of mine, Malin DeLong, graduated from Harvard Medical School and went to the NIH during the Vietnam era. And he was interested in the basal ganglia, which disorders of the basal ganglia are often expressed as movement disorders. Parkinson's disease, Huntington's disease are basal ganglia disorders. Dystonia is a basal ganglia disorder. And his hypothesis was that we knew so little about the basal ganglia that if he just recorded the neurons in a monkey that was just sitting in a chair or moving, that he'd learn something interesting. Now, I can tell you that this is an absolutely unfundable hypothesis today. The notion that you could discover something interesting just by exploring. So he published a paper in 1971, and the figures from that paper are on most neurosurgery operating room walls because they guide you to where you have to put deep brain stimulating electrodes in the brain. It's the foundation, the critical foundation for deep brain stimulation. So in 1971, he publishes this paper, and in the course of doing the research, he discovers a set of neurons at the base of the brain and what's called the nucleus basalis. And it turned out that Malin was in a carpool. He lived in Columbia, Maryland, and every morning commuted with Don Price to Hopkins. And Don was an expert in Alzheimer's disease. And they looked at the brains of Alzheimer's disease patients, and they saw that these neurons in the nucleus basalis degenerated. And Malin's experiment wasn't about the nucleus basalis, but he discovered these neurons just in the course of doing the research. One of the few treatments that we have for Alzheimer's disease is using cholinergic drugs to replace what happens when the nucleus basalis neurons degenerate. It's not a great treatment, but it's one of the few that works. So that was really 10 years after he did his recording experiment. Then 15 years later, there were a group of kids who were making synthetic heroin in their basement. And one of the contaminants was a drug that is abbreviated MPTP. And these kids, MPTP is a selective neurotoxin. And these 21-year-old kids were ending up in Bay Area emergency rooms with Parkinsonism. And no one could figure out what was going on. And it ends up that MPTP is a selective neurotoxin for the basal ganglia. And when Malin learned about this, he used MPTP in monkeys to create a monkey model of Parkinsonism. And because he, in the 70s, had recorded in the basal ganglia, he could go back in the 80s and record in these Parkinson monkeys and find out what was changed. Where were neurons that were hyperactive? Where were neurons that were inactive? Where were neurons showing abnormal activity? And he published a really seminal paper about that. And one of the areas that was abnormally active was the globus pallidus. Now, the globus pallidus, I'm sure, is familiar to all of you. In Latin, it means pale globe. It's the major output nucleus of the basal ganglia. And the globus pallidus, one of its major inputs is the subthalamic nucleus. And this region, basal ganglia is a lovely term. It's in the base of the brain, all right? The subthalamic nucleus is the one place that if you put a lesion there, you can reproduce a basal ganglia disorder. And the disorder you reproduce is hemibalism. And what is that? Somebody will have abnormal flinging movements of their arm or their leg. Well, the subthalamic nucleus is a major input to the globus pallidus. And Malin reasoned that if he lesioned the subthalamic nucleus, he might normalize the globus pallidus activity and cure some of the symptoms of these Parkinsonian monkeys. No study section would ever have approved this. They would have said, you have a Parkinsonian monkey and you're going to put a lesion in a place that causes hemiballistic movements? This is crazy. We did the experiment and it worked. Monkeys that were extremely Parkinsonism were normalized. And then he took this to humans, and subthalamic lesions help Parkinsonism. And then, along with Benabid in France, instead of making a lesion, they put in deep brain stimulating electrodes. And it's an effective treatment. When L-DOPA and other treatments begin to fail, deep brain stimulation of the globus pallidus and the subthalamic nucleus are effective treatments for treating Parkinsonism. So Malin got the Lasker Award, which is one step before the Nobel Prize in 2015. So I want you to really understand the time course here. The first critical paper that was the foundation of this work was 1971. 1971 paper led to insights not only for Parkinson's disease, but for Alzheimer's disease. And when he did that 1971 experiment, the hypothesis was, if I explore the basal ganglia, I'll find something interesting. That's it. And we don't have those kinds of experiments going on anymore. The funding is so bad that we have cut out the opportunity for discovery. Now, neither one of my sons is an active neuroscientist. No one would recommend their kid go into neuroscience when funding levels are as low as they are. So not only have we cut off the opportunity for discovery, but we've cut off a new generation of neuroscientists. Who would go into a field where your chances of funding are 10%? So my sermon about this is nearly over, but it's us that have to change the situation. If we want cures and treatments for some of the diseases that are at epidemic proportions, the opioid epidemic, the Parkinson's disease epidemic, the Alzheimer's disease epidemic, if we want to make progress in this, it's only by new discoveries. The discoveries, the information now to treat these diseases doesn't exist. So polio is a wonderful example. No one here suffers from polio. We never cured polio. We vaccinate against it. Rabies, if somebody contracts rabies, they die. We vaccinate against rabies. So maybe we need to vaccinate against Alzheimer's disease or Parkinson's disease. We have to have new approaches, new ways of looking at these things. I'm told that if we could stop mothers drinking and smoking in their third trimester, that we would have a profound impact on our educational system. Well, getting people to give up nicotine is one of the most difficult drugs. And getting people to all of a sudden give up alcohol is also near impossible. And so maybe we have to vaccinate. We have to protect our kids. So the moral of this story is, first, the time course of discovery requires foundational work that sometimes is 20 to 25 years off. If we want to have an impact 20 years from now, it's because of the research that's being done today. And second, we've cut off the opportunity for discovery, and it's really important for us to buy a ticket. Thank you.
Thank you very much for the talk. We all appreciated it. I have a question. When I was a young child, I was left-handed, and my aunt told my parents, don't have your kid left-handed. He's going to severe problems later on, et cetera, et cetera. So what happened was my mother and my aunt made sure that I became right-handed by tying my hand behind my back, doing all sorts of things like that. And it turned out now I do everything with my right hand. I don't do anything at all with my left hand, except the only thing that I've noticed that I do with my left hand is in case of an emergency, I instinctively, like if I'm going to fall or something, I will put my left hand out first. Otherwise, everything else is right-handed. Is that literally hardwired into the brain, or how?
Well, it's a fantastic question. My father went through the same experience. He was left-handed, and his mother tried to change him. And when I talked to my grandmother, she said, your father always wanted to do things a different way. What we know now is, in fact, that early on, you develop connections that are activity dependent. And so the fine control of the fingers is something that you develop. It's not present at birth. It develops postnatally. So how you use a pathway has a very big impact on its development. I was going to do a demonstration showing the acquisition of motor skill, because that's also something that you can acquire. Mm-hmm. And the issue that I think you've raised is the connections change when we acquire a skill. And that's a continuing challenge for neuroscience. I can teach you how to juggle. I learned how to juggle as an adult. We can all learn new motor skills. So what happens in the brain when we acquire those motor skills? And I think that's what your question's getting at. Yeah.
I'm wondering, given your critique of funding opportunities, what you think about this new generation of philanthropically funded science, such as the Chan Zuckerberg Initiative, commitment to something called the BioHub. There are reputable scientists leading it, Steve Quake, Joe Derisi. Are you talking to them about changing the model for early stage discovery?
So it's another great question. And part of the answer is I think that any additional money into science now is going to have an impact. But all too often, there's a notion that it's research at Harvard or Stanford or MIT that's going to cause a difference. But discoveries come from everywhere. And I think that if you look at people who've had the Brain Initiative and the people who had the Zuckerberg Initiative, they come from the places that you'd expect, and they're not going to fund research all across the country. Yeah.
I recently became aware of a program offered at a University of Pittsburgh under the direction of Dr. Lopez from the Department of Neurology. It's called BRITE, B-R-I-T-E. And the premise is that they can grow white matter in the brain for people with early Alzheimer's disease by the use of a program that works with people to teach them to play the marimba, to do yoga and exercise, to do computer games, and to do artistic things. I'm wondering if you've heard of that and what you think about it.
So I've heard about it. I think it's fantastic, and that there's real science behind it. So at one level, if you look at the brains of experts pianists, and you look at their hand area, and you look at, say, an ordinary pianist like me, their hand areas are larger. The cortex is plastic, it changes and grows, and what grows are the connections. And so the notion that Bright is using what are normal mechanisms to promote recovery, I think is terrific. If you go on the net, there is a fantastic video on YouTube of a patient with extreme Parkinsonism and freezing of gait. And he told his neurologist that he could ride his bike, and the neurologist didn't believe it. And so brought him his bike, and this guy goes riding off into the sunset. This is a fellow who was absolutely frozen in spot, couldn't move at all, would fall down, was having difficulty just standing upright, gets on his bike, rides off into the sunset, turns around, does a lot of predictive things that require body adjustments that normal, this is an 80-year-old, fantastic, hops off his bike and then is frozen in place. Completely frozen in space. You just type into Google Parkinson, freezing of gait and bicycle riding, and this video comes up. It's spectacular. So we think we know the pathways that enable that there are ways of compensating. There are systems that aren't impacted. And in Alzheimer's disease, there are a number of instances where people all of a sudden have this sort of breakthrough phenomenon where they show abilities that they haven't shown in a while. And looking at what are the capacities for other systems to compensate, both through normal plasticity and choosing alternative routes, I think is very exciting.
Thank you.
Could you discuss, please, the theory that stress can suppress our immune system and therefore lead to disease, and ways that people can help boost their immune system so that it helps keep them healthier? That seems to be a pretty wide theory. Do you agree with it? And
Yeah. So if you asked me that question 2 years ago, I would have said, this is nuts and granola. Speak to my sons. But I think the evidence is growing, and when I'm not here speaking, I'm writing a grant to look at that specifically, where we're going to look at the cortical areas that control organs of immunity, like the thymus and the spleen. And what we know about the neural control of them is that those pathways are immunosuppressive. when they're active. And so what cortical areas? Are they areas of conflict? Are they areas involved in stress and depression? That when we're stressed and depressed, that their output is immune suppressive. And then once we know that, then there are strategies that one could take in terms of therapies. So again, I don't think that we're talking about pathways that cure cancer, but that they would have an impact, absolutely. And the evidence about depression and cardiovascular disease is getting more and more compelling. It is one of the major risk factors for cardiovascular disease. We have a clue now about what areas are critically involved in depression. People are using deep brain stimulation and also transmagnetic stimulation to influence the function of those areas. And I think those are new therapeutic approaches to deal with the brain's control of the immune system. So I think it's real, but for me, it will be more real once we know the pathways. That's what's going to be critical. Yeah.
My name is Rob from Florida. I have 3 quick questions. Just yesterday, it was announced by Elon Musk that he is beginning either a company or a study on the interfacing of computers with the brain. That's number 1. Number 2 is, have you heard of Dr. Nussbaum? He's in the Pittsburgh area, and his study in spirituality as far as healing. Need a comment on that. And the third one is, about 4 years ago, there was a book, I can't recall the name, I think it was on plasticity of the brain, maybe Dr. Schwartz, but he was able to overcome OEC, obsessive compulsive disease, by changing the plasticity of the brain.
So let me get to your first question. This issue of Musk using brain-machine interface work. This happens to be where the University of Pittsburgh is a leader in that field, and a colleague of mine, Andy Schwartz. And I like to talk about this example because it shows the importance of discovery. So in the 80s, Andy proposed to implant a monkey with an array of electrodes and train the monkey in a virtual reality to have the signals from his cortex control a robotic arm so that the animal would feed himself. And he sent this to a conventional NIH study section. They said, this is absolutely ridiculous. Okay, a monkey in a virtual reality, how is the monkey going to know what to do? Okay, you put virtual reality glasses on a monkey, this is ridiculous. And then the signals from the monkey's cortex, the monkey's going to know how to control the robotic arm and feed himself, forget it. So 7 years later, the demonstration that Andy could do this was on 60 Minutes. Then they took that demonstration, and with some DARPA funding, they implanted a woman who was paralyzed from the neck down, and she had one goal. She wanted to feed herself some chocolate and put an array of electrodes into her motor cortex, and they used the signals to control a robotic arm. And within under a month, she was able to feed herself chocolate. And that was also on 60 Minutes. So the next step in this is not just to use the signals to control a robotic arm, but if somebody's paralyzed, they've got a normal arm there. To actually implant muscles with optogenetics, another technique, and have them control their own arm. So Obama came to the University of Pittsburgh, and the second subject who was implanted not only had the ability to control their arm, but they took the robotic arm and they put sensors in the tips of the robotic arm's fingers, and they had that feedback to the sensory cortex, so the subject could feel what the robotic arm was touching. And that's critical because he was going to do a handshake with Obama. And if he squeezed the robotic arm too quickly, he would have broken his hand. Okay? But he had a successful handshake, and the fist bump is on the net. So it's these kinds of things. I should say that Andy Schwartz is not funded now. And why? Because the research is just too creative. You say, he can't do that. And you say, well, look, after the third time, you said he couldn't have a monkey work in virtual reality. You said you couldn't have the monkey control a robotic arm. You said you couldn't have the woman feed herself chocolate. You said you couldn't stimulate the sensory cortex and give someone feedback from the robotic arm. He's not funded today. So there's something very wrong with the system. Yeah. My answer was too long, so I don't remember your other questions. But, hope that's helpful.
The first one?
Your second question.
It was Elon Musk? No. The third one. Okay. It was the book written on plasticity of the brain, and how in the book he related how he was able to cure his patients of obsessive compulsive disease. That's where well, you know what that is. Mm-hmm. And he was able to overcome it.
I'm not familiar with the book. Sorry. Yeah. Okay. But I think one of the outcomes of the kind of research we're doing is that by identifying areas, by looking at networks, it gives you insights into approaches. I like to use the analogy that suppose you take your car to a dealer to have the car fixed. And the dealer says, I only have the wiring diagram for a Model T. He said, well, I don't have a Model T, I've got a Ferrari. I want my Ferrari fixed. Okay, well, I'm going to use the Model T, okay, as the basis. So what we have the wiring diagram for worms, and we have the wiring diagram for sea slugs, aplysia, and we have the wiring diagram for some part of the mouse nervous system. We don't have the wiring diagram for the primate nervous system. So is there any wonder that our repair shops can't fix our brains? Yeah. Thanks.
Thank you.
Thank you. I was tempted to do two questions, but I'll do one. One of your predecessors about 60 years ago at Pitt transferred there and was getting his budget put together and was on the road to a big discovery to protect us from an important disease. And to make progress, he had to get some primates to do the research on. The monkeys in 1954-55 cost $30,000 apiece, more than his lab assistants or professors. But Dr. Sabin was able to get the monkeys and protected us. Is there a problem today in doing research because we, for ethical reasons, are limited into what we can do and what we can study?
Yeah, so absolutely, we can't do any research on chimpanzees. It's been outlawed. I think people might agree that we shouldn't do research on chimpanzees. But what about a dying chimpanzee? What about an animal that's clearly needs to be sacrificed because it has some cardiovascular disease? So I wanted to do that because I felt that we need the wiring diagram of our brains. It's very simple. And so I learned of a chimpanzee that was terminal stages of disease and contacted the primate center, and they said, we'd be very happy for you to take this chimpanzee off us because it's costing us an enormous amount of money, but the animal rights activists would burn down your house. Right now, we can't, by law, do any research on chimpanzees. And animal rights activists are hoping that they can do the same thing for macaques, which are the major laboratory animal. And there's a famous statistician who said, all models are wrong, but some are useful.
Right.
And I would add to that that some models are more useful than others. So right now, with 94 to 96% of the research being done on rodents, we know a great deal about the rodent brain. But I don't have any difficulty in telling everybody here that the rodent brain is different from mine. Okay? And that's key. Okay? MTTP, that Parkinson model, doesn't work in rodents. Okay? Rodents don't have schizophrenia. There are no decent rodent models of Alzheimer's disease. There are rodent models of ALS. The 20 clinical trials that have been done developing the drugs that have been used in those models have all failed miserably in humans. And so if we want to make progress, it's because we use the best possible models. Yeah.
And we are protected from polio today.
Yeah.
A lot of your examples today were about training of the motor system. In fact, I hope you're going to straighten that kid out before he drives home tonight. But what do you think of the commercial stuff that's out there that supposedly trains the cognitive system and folks like us who are really worried about losing our cognitive acuity? There's an awful lot of that stuff out there. A lot of it claims to be based on research funded by NIH. What's your take on that?
So if you use something, there's no question that it improves. There's just overwhelming evidence. And some of the best actually came in the 1950s. They looked at Cuban women rolling cigars. And they are experts after they roll 1 million cigars. And they looked, do they get faster when they roll 2 million? Yes. Do they get faster when they roll 5 million? They get faster still. Do they get faster when they roll 10 million? Absolutely. And so you'd say, look, after 1 million cigars, these people are experts. But it's really, a lot of practice leads to incremental improvements in performance at almost any age. I lived next to the principal harpist at the Pittsburgh Symphony, and she practices 3 to 4 hours a day. I say, don't you know it by now? And she says, to keep her edge, she needs to practice. Okay, and so all of us need to practice, as I think. So those programs that lead us to practice and lead us to use our brains, I think, are effective programs.
Do they generalize, or are they just for the task you're doing?
Do they?
Do they generalize beyond the narrow task you're doing in the training?
Right, and so that's a great question, and what I think I demonstrated is what you practice is what you improve, and there's not much broad generalization. It's a great question. Yeah.
Both in the newspaper and one of the questions here, you addressed immunology somewhat. My wife has lichen sclerosus, and it's a condition that allows, because it affects the immune system, cancerous cells to grow. They're talking about using T1 inhibitors as a way of correcting the condition. You talked about stress. I didn't think she was in a particularly stressful environment, but is there other alternatives that you know about immune system that might be good things to look into?
So one of the things that I'm acutely aware of are the things that I don't know anything about, and this is one of them. And so I couldn't make any useful comments. I'm sorry.
Okay.
Yeah.
Hello. There's a lot of publicity lately about gene editing and using technologies like CRISPR. Do you see any application there for brain health or overcoming some diseases like Parkinson's?
Yeah, so absolutely. I mean, and two really fundamental applications. First, we know the gene that's disordered in Huntington's disease. So CRISPR could repair it. But then we get into all kinds of ethical issues about manipulating human embryos. But I think this is something that we need to really discuss. But the other issue is we have no decent animal models of ALS, Huntington's disease, any number of neurologic diseases. And we're using CRISPR to try and create non-human primate models of human disorders. And I think CRISPR is going to offer incredible opportunities. It's very expensive because you have to implant embryos into monkeys, and there's a very low rate of success. And so to do this, you need 200 females that are housed with vasectomized males. And so it's an expensive process. But it's key that we have non-human primate models of human disorders, because I think when you look at the progress we've made in the treatment of Parkinson's disease, in large part, deep brain stimulation depended entirely on the development of a non-human primate model.
Yes, thank you for your information. I couldn't agree with you more that we need a lot more funding for this. But in the meanwhile, I do have Parkinson's, and I'm in really quite good shape because of a wonderful doctor at Cleveland Clinic, and also because of the great amount of exercise that I try to continually have. And I get information through the Michael J. Fox Foundation, and a lot of this information is very helpful for some of the hidden things of Parkinson's, where most people think of Parkinson's just as leg problems and arm problems. There's a lot more than that. And I find these webinars that they give to be very, very helpful. Anything we can do to increase the funding would be great. I've been in a lot of studies, but that's best I can do.
No, but that's super. I had the experience of spending some time with a Parkinson patient and asked, what was the disability, the symptoms that caused them the greatest difficulty? Was it the arresting tremor?
For me?
And the patient said it was constipation.
Oh.
That was the major, that was the
Yes,
major
that's
issue
one
for
of
them. And so it's part of the reason why I've looked at this brain-body problem, is because that system is influenced in Parkinson's disease as well as many others.
Oh, yeah. And trouble sleeping and depression and a number of other things, yes.
Yeah.
Yeah.
I had a mini stroke in 2013, and it turned out that a blood vessel broke in my head, and it filled my head with blood. So I came out of the stroke, and it didn't affect me physically. It was fine, but it affected me mentally. And I have a very hard time retaining any information that comes my way. I have many friends here in Chautauqua, and I will pass them on the street, and I know who they are. I know they're a friend. I can't remember their name. I don't know where I know them from. And I am very disturbed at this problem that I have. And I'm told something that I wanted to remember, like somebody's name or whatever, and 5 minutes later, it's gone. I cannot remember. Is there any exercise or anything that I can do to help get my memory back the way it was? Well, maybe not the way it was, but at least better than it is now.
Right. Obviously a great question, but the answer should come from my colleague, Larry Wexler, who's a stroke specialist. He's chair of neurology. Maybe, Larry, you'd stand up. Larry's the chair of neurology and the head of the Stroke Center at the University of Pittsburgh, and he'd have much more insights about that than I would. So thank you for your question, but maybe you should talk with Larry afterwards.
I'm sorry, I don't understand. Thank you.
Thank you for the interesting lecture. We have a 6-year-old son who has autism, and we're thinking that's a function of the brain. Do you know what the probability is that he might outgrow this?
It's another great question. And that's not my area of expertise. So what's fundamentally is what we know about autism in terms of it's a spectrum, and whether he's able to compensate for the difficulties he has. That's really where on the spectrum he is, and what are the specific issues in terms of compensating for functions. So I couldn't be specific about a particular individual.
Okay, thank you.
This is a lighter question, but can we sometimes hear our brain work? When I was in my early 40s, I had to study algebra and calculus, and it was a very intense effort, and I could swear I could hear my brain crackle.
Yeah. Well, based on your experience, we can all hear our brains work. Yeah.
Do you have some other examples, or am I the only one to mention that?
I don't know of other examples.
Okay.
One thing is that I'm very well aware of the brain-mind-body connection, having visualized the tumor away with LaShawn. But my question is germane to this audience. If you look around, we can see that we're of a certain age. And one of the things that the public doesn't hear about are the developmental processes of the aging brain. Can you recommend a book? Can you recommend something that all of us here can learn about what to expect as we go forward?
Oh, it's a fantastic question, because I've wondered the same thing. We're probably of the same generation, although I look a little older.
I'm definitely older.
And I don't think there is the single resource. And so I've thought in my spare time of trying to create it.
I'm starting a website to talk about that along with funny rhymes. So that's what I'm looking for.
Yeah, I don't know the resource. Yeah.
Thank you so much for a very stimulating presentation. Can you tell us a little bit about the current understanding and knowledge of the connection between the microbiome, our bacterial population in our gut, and its relationship to the brain?
This is a terrific audience. What a great question. In fact, we know more about how the microbiome can influence the brain, in some cases through inflammation. What we don't know is the other direction, how the brain influences the microbiome. And it's a handshaking relationship. And we've ignored this brain influence on the microbiome. So what I'd say, in my spare time now, I'm writing a grant on just this issue, because when I looked into what were the origin of my stomach aches when I was a kid, there's no understanding of the regions of the cortex and how we move, think, and feel, and how it influences the gastrointestinal system. Where, in fact, for the cardiovascular system, there's a similar gap, but we have a much better understanding of how important it is because strong emotional stimuli, both positive and negative, can lead to sudden unexpected deaths due to heart attack. So we have clear evidence that strong descending inputs from the cortex can cause left ventricular heart damage that lead to sudden unexpected death. So for the microbiome, I think it's going to be just as likely that how we think, how we feel, how we move has an impact on the microbiome. And it's unexplored, totally unexplored.
Thank you.
Okay, last two questions.
Thank you for coming today. Could you talk a little bit about the notion of left brain versus right brain and, you know, people having different skills because of it or better abilities?
Not in 2 minutes. I mean, it's a very long, an interesting topic. Yeah, I couldn't give a quick answer, I'm sorry. You know, yeah, I happen to be a right brain person, and that means I can't give a short answer. Okay.
This is quick. We've all been sitting a lot for this past week in various lectures and things, and I keep reading that sitting is the new smoking, and we should all be moving more, and that all of that affects how you age and so forth. Did you want to comment on that or no?
No, I think this issue of core exercises is a very broad one. So preliminary studies that we did with injecting the heart and looking at what cortical area has the most direct and largest control over the left ventricle, it was the motor cortex. And again, axial body representation of the motor cortex. And in addition, those cortical areas that are involved in planning and in decision making, they're the ones that control the heart. Now, you know, when we move, we don't think, well, the first thing I have to do is increase, the cardiac output to support. But if we don't change cardiac output predictably when you stand up, you have a hypertension that makes you collapse. And so there's a certain housekeeping that has to go on every time you move. And, basically, the motor system has two jobs. It has to move the limbs and support posture, but there's a whole lot that goes on inside in terms of your internal organs that you don't intentionally say, well, I've got to adjust blood flow to my kidney, and I've got to, stop the blood flow to my stomach, and I have to do this and that. The nervous system takes care of that for you. But these are powerful influences.
Movement.
Yeah, absolutely.
Thank you.
Thank you.
With those words, we should all stand up and move, I think. Thank you so very much for your great lecture today.